So thank you, Rainer. I would like to start by congratulating you with the opening of the new center. I think it's really fantastic to see what you and your team have been building up here in Maastricht in the last 10 or 15 years. So very many congratulations for that. So today I want to talk about the thing that I'm really interested in, and that is the question, how the visual brain constructs objects from features. And I'll simply start with the take-home message. So if you see a picture like this, your visual system is confronted with a lot of information. And as a first step towards image comprehension, it's really important that the visual system groups together all those features that belong to one of the objects, in this case, a small bird, and to segregate those features from features that belong to different objects in the background. And the take-home message is that this is accomplished by the visual system through what we call an incremental labeling operation, where eventually all the image elements that belong to a single perceptual object are labeled with enhanced activity in the visual cortex. This is what psychologists call object-based attention. And we simply want to know how object-based attention works. Okay? So if the picture that comes into the visual system is this, then the visual brain constructs a labeled image where all those features that need to be grouped are labeled with an enhanced response. So in my talk, I'll first briefly introduce to you the two concepts of feed-forward and feedback processing. Then I'll be applying those ideas to a texture segregation task, and I will talk a little bit about the detection of boundaries and the grouping of image regions. Then I'll say a few words about the role of the different cortical layers. And in the last part of my talk, I would like to share with you some new data that we obtained, actually in humans, on the effects of imagery in visual cortex. Okay, so let's talk about feed-forward and feedback processing, and this is a slide that I guess most of you have seen many times. This is the not infamous study by Velleman and Van Essen, published many years ago, that gave colors to all those areas in the macaque visual system that are involved in vision. So neurons in all these different areas are involved in different aspects of vision. And what we and many others would like to understand are the messages that are being exchanged between the neurons in all these areas while we try to make sense of what we see. Now, these messages are being exchanged through this dense network of cortical-cortical connections. This is from the same study. And what you see here is that the topology of these connections allow you to arrange all these areas into this hierarchical scheme where the retina is at the bottom of the hierarchy, and then you have all these different higher visual areas up here. Now, if you present a new image to the system, the first thing that happens is that you have all these feed-forward connections that propagate information from the retina to higher visual areas. And the processing, we believe, that is carried by this first phase of feed-forward processing, we sometimes call it the feed-forward sweep, roughly corresponds to what psychologists call pre-attentive vision, vision without attention. This is then followed by a phase in which feedback connections, here in blue, and also lateral connections come into play. And these connections allow for a recirculation of activity. So now neurons in, say, area 46 of prefrontal cortex can take influence over the activity of neurons down here in primary visual cortex. And it's the idea that during the second phase, where the recurrent processing starts to occur, that roughly corresponds to what psychologists call attentive vision, vision with attention. Now, if you make neural networks, and we sometimes do that, and it turns out that you can start to explain already quite a lot of what's going on in the visual system if you only put in the feed-forward connections. So you can, for example, explain why receptive fields at higher hierarchical levels become larger, because these neurons get information from a set of neurons in lower areas with displaced receptive fields, so they can pull information about, from a larger region of visual space. Now, if you choose the connections carefully, you can also explain why tuning in higher visual areas is more complex. So if you record from a higher visual area, such as the inferior temporal cortex, you might encounter a neuron that is tuned to a very particular shape, say the shape of a bird. And some of these neurons might even be picky about the precise color of the bird. So you might even find a neuron that only responds to birds that happen to be brown. Okay, so in some sense, these are already sorts of groupings. And we think that from these, what we call base groupings, the more incremental groups are being formed. But for that, you need the feedback and lateral connections, here indicated in blue and red. Now, having these connections is going to tremendously change the processing you can expect from your network. Because now a neuron say in the primary visual cortex with a small receptive field, can, through a sequence of lateral connections or through a sequence of feed-forward and feedback connections, also get information from outside its classical receptive field. Okay, so in such a scheme, neurons become sensitive to information outside the classical receptive field. Now you have to realize that the shortest route from the retina up to a cell at a certain hierarchical level, the shortest route only uses feed-forward connections. So if you want to see these recurrent influences that involve lateral connections or feedback connections, you have to wait a little bit longer because they will be associated with additional synaptic and propagation delays. So in such a scheme, neurons will convey different messages at different times. So what now happens if we present this bird? The idea is that then in the first feed-forward phase, the growth shape of the bird is being detected in feed-forward processing, and then through feedback connections, extra activity is propagated to all those cells that respond to features that belong to this object. So in this case, a neuron that would be responding to this edge might have extra activity because it's found on this bird. So that's the idea. Now this is a slide that is, I think, common in the thinking of many neuroscientists. It has been expressed very elegantly by Sherman and Guillory many years ago. And the idea is that feed-forward connections tend to be driving. They activate the cells, determine the receptive field properties, whereas the lateral and feedback connections tend to be rather modulatory. That is, if a cell, by the driving connections, is brought to fire 40 spikes per second, the modulatory connections can change it into 60 spikes per second or into 20 spikes per second. But they tend not to drive cells. So I think that is true for most of the things that I'll be presenting. But there are actually, as there is the case for every rule, there are some exceptions. And I would, towards the end of my talk, also like to talk a little bit about one of these exceptions, which is related to imagery. Okay, now let's go to the task that I will be talking about in the main part of my talk, which is this texture segregation task. Okay, so I guess that we can all see that there's a square figure that is actually composed of elements of one orientation that may even seem to be floating on the background of another orientation. Now, already many years ago, a colleague of mine, Victor Lamme, demonstrated that neurons in primary visual cortex care about this perceptual organization. So what he did here, he recorded from V1, and this is a receptive field of a group of cells, or maybe a single neuron in V1. And you see that there are some image elements in the receptive field that belong to a figure. And here, he carefully kept the image elements the same, but now they belong to a background. And what he demonstrated is that the initial responses, as you can see here, are mainly determined about what's going on in the receptive field, but after a delay, you see that you get extra activity if the receptive field falls on the figure. So yes, neurons are influenced by the information outside the classical receptive field, and you can also see that they convey different messages at different times. So here you see this labeling process that I was talking about in action, and this is the process that we really would like to understand. Okay, so how does this work? So it's a good idea then to look into the psychological literature, and you'll find that psychologists typically propose that there are two mechanisms for figure-ground segregation. And the first process is what they call a boundary process. That is a process that is sensitive to those feature discontinuities that happen at boundaries. So that's boundary detection. And the second process is region filling or region growing. And the idea is the following. Suppose that you select some image elements here in the middle of the figure with the left orientation, then you might want to co-select adjacent image elements of the same orientation because they're likely to belong to the same perceptual object. Now we want to understand mechanism, so then it's a good idea to again look into the computational neuroscience literature. And what you'll find is that people typically propose that for boundary detection, what you need is iso-orientation inhibition. So neurons that are tuned to the same orientation should inhibit each other. Now, how does that work? Well, suppose you're a neuron and you have a receptive field on the background and you're tuned to the right orientation. Then all your neighbors are very active and they're going to provide you with very strong inhibition, thus suppressing your response. Now, the same will happen if you are a left-tuned neuron in the center of the figure, because also in that case, all your neighbors are going to provide you with strong inhibition. only if your receptive field is relatively close to a boundary, the inhibition comes only from one side, okay, causing a relative release from this inhibition. So this gives you boundary detection. Now region growing. Now the remarkable thing about region growing is that it needs the opposite connection scheme. It needs iso-orientation excitation. Now how does that work? Well, suppose there's a response here in the center of the figure to the left orientation. Those cells, for some reason they start to enhance the response. Maybe there was a cue at that location. Then they want to tell their neighbors that they should also enhance their response if they're tuned to the same orientation. So the response enhancement will spread, of course, until it reaches the boundary, because we did not put excitatory connections between neurons tuned to different orientation, and then this propagation will stop at the boundary. Okay? So we see that we have a small problem here, because we want to make a neural network, and for boundary detection we want to put in iso-orientation inhibition. For region filling we want to put in iso-orientation excitation. If you're going to make a neural network and don't think about it, these effects of course will cancel. Okay, so we need to come up with something better. And the idea that seems to be most consistent with the data that we obtained is that boundary detection is actually closely tied to feed-forward processing, whereas this region filling process depends more on the feedback. Okay, now you can make a neural network model that establishes this, and I'm going to show you how this would work. So this is now primary visual cortex. This is the stimulus with the left figure on the rightward background. Here is a map of visual space with neurons tuned to the left orientation, here neurons tuned to the right orientation. Those guys respond to the figure, those guys to the background. Of course, in reality, these two sets of neurons would be intermingled in a single retinotopic map. And we put in the feed-forward pathway the iso-orientation inhibition scheme for boundary detection. And yes, we get extra activity at the boundaries here and also in the background representation. If we add the two maps together, you see indeed, yes, you have extra activity at the boundaries. This is what you would have predicted. Now the trick is that we're going to do this not only in V1, but we're going to repeat it in higher areas as well, where receptive fields are larger. And this is the result of that idea. So in primary visual cortex, you can see that in the center of the figure, there's still the suppression going on. But if you go higher, receptive fields become larger and larger, and at some level of the hierarchy, you reach an area where receptive fields have about the same size as the figure. So now you have a neuron that is tuned to the left orientation. His neighbors with different receptive fields tuned to the left orientation, they are actually have their receptive fields on the background. So this guy in the center receives very little inhibition. Okay, this is called pop-out. So in the feed-forward pathway, we put in boundary detection, or we get boundary detection and pop-out at different spatial scales. Okay? And now we're going to use the feedback, the iso-orientation excitation for region filling in the feedback pathway. Okay, and if you make sure that these connections are indeed, say, between neurons tuned to the left in the higher areas that go to neurons tuned to the left in the lower areas as well, then you can make this work in a neural network. So, and this is quite consistent with the idea that boundary detection and pop-out is pre-attentive, it is stimulus-driven, and the prediction of our model is that the region filling should depend more on the workings of visual attention. Okay, so a prediction now becomes that boundary detection is an early process, is stimulus-driven, and the region filling happens a little bit later because it depends on the loop through the higher visual areas, and it should be task-dependent. Now, to test this, we recently did a study in which we had monkeys looking for this square figure on the background of a different orientation on some of the days. These were the days that they were supposed to attend the figure. On different days, they saw precisely the same stimulus, but now they were supposed to trace one of these curves. Okay? Now, on figure detection days, they were supposed to make an eye movement to the center of the figure, and on curve tracing days, they were supposed to make an eye movement to the end of one of these curves. And this would be the receptive field, so it's smaller than the figure, and we also varied the location of the figure relative to the receptive field. So on some trials, the receptive field would be on the center of the figure, could also be on the edge, or we would be falling on the background. And this then shows you the activity, and we basically replicated what I now call the Lamé effect, so this figure ground modulation. So you get extra activity elicited by the figure as opposed to the background, which is this red curve. Now people sometimes say, well, this seems to be a relatively subtle modulation. It's true if you compare it to the peak response, but you have to realize that by the time that this modulation occurs, these peak responses are no longer there. And then we're really talking about almost a doubling of the response. So we want to know the spatial profile across the figure of this different signal. So we're going to subtract the red response from the other response. And this is done here as a function of space and time. So what you see here on this axis is the location of the receptive field relative to the figure. So here the receptive field's on the background, here it's on the first edge, here it's in the center of the figure, here it's on the second edge, and here it's on the background again. And this is time. So this is what you see on the x-axis is this modulation, right? This difference response. And you see that the difference response occurs earliest at the two boundaries, and that you then later on see a filling in of the center of the figure with enhanced activity. So this is quite consistent with the model that I've just proposed. Now this is the situation when the animal was interested in the figure, and this is the situation when the animal was tracing curves. He was ignoring the figure. And what you can see is that you still got the boundaries, but the filling in process was much less complete, consistent with the idea that indeed this boundary detection process is stimulus driven, and this region filling process depends indeed more on the task that the animal is doing. Now we also recorded an area V4 at the same time, and in area V4 receptive fields are larger, and you see that now you'll have the response enhancement also early on in the center of the figure. And we think that this is early enough through the iso orientation excitation to help V1 with filling in the center of the figure as well. Okay, so let me summarize this. So what we find in this task, so if this is V1, which is a map of visual space, we present the stimulus, then neurons are driven by the stimulus because there are line elements everywhere. Early on you get extra activity at the boundaries, irrespective of whether the animal is interested in the figure or not. Then you get a filling in of the center of the figure with this enhanced activity, but mainly so if the animal is interested and less so if he's directing his attention elsewhere, as if those unattended objects are left in a somewhat more primordial state. Okay, now I would like to say a few words about the role of the different cortical layers in the same task. Remember that we're trying to carry out two different computations, iso-orientation inhibition, iso-orientation excitation. So we thought it might be a good idea, the cortex has these 6 layers, it might be a good idea if some of these computations are kind of separated into the different layers. Now actually, to be honest, although we've been studying this for many years, we don't have so many ideas of why the cortex has 6 layers and what is the specific processing that's being carried out by these different layers. Okay, now we know from anatomical studies that feed-forward and feedback connections are separable. They terminate in different cortical layers. And this is actually a cytochrome oxidase staining, but it highlights nicely the layers where the feed-forward connections terminate. So they tend to terminate in layer 4 and layer 6. And this is an anatomical tracing study where they injected tracers in area V4, and you see that the terminals of these feedback connections tend to be in layer 1 and in layer 5. Now what you see down here is actually subcortical white matter. So we have to concentrate here on layer 1 and layer 5. And then we started to use these laminar probes that were actually developed by Charlie Schroeder and that allow you to record from all the layers at the same time. Well, the animal is doing the figure ground task, so he's simply supposed to make an eye movement to the center of the figure. And we're interested in the laminar profile of the feed-forward response and also in the laminar profile of the feedback-driven modulation. So this is another replication of the Lambda effect then again. So a visual response and then the figure ground modulation, so more activity elicited by the figure as opposed to the background. We also had a condition in green where the entire screen was filled with bars of the same orientation. So let's have a look of the peak response across the cortical layers. So this is shown here. So this is superficial layers, this is deep layers, and this is time. And you see that indeed activity starts in layer 4 and layer 6, the terminals of the projections of the LGN. This is shown the same thing at a somewhat higher magnification, and yes, first activity in layer 4 and layer 6. Now, I was somewhat surprised by this, but it turns out that layer 5 is then to follow, and layer 2 to 3 follows even later. Okay, so this doesn't seem to be compatible with what is known as the canonical microcircuit. Now, this is not new. You could have known this had you looked into the literature. John Mounsell had a paper like this, and also Jean Bouillet has shown this. Okay? Now, the nice thing of these laminar probes is that you can also look at what is known as the current source density. And in principle, it works as follows. If there's excitatory input into a cell, the current flows into the cell, and this gives rise in the current source density to what is known as a sink, which is shown here in yellow and red. And then the current is in the cell and has to go out somewhere else, and then it gives rise to a source. So sources are indicated here in blue. And it has been known from previous work from Charlie Schroeder and from Uwe Miedsdorf is that the early sinks that give rise to the visual response actually happen in layer 4, and this is nicely replicated in this study. What was not known is what is the laminar profile of this response difference, this modulation. Right? So we're going to look at this different response now as a function of laminar depth and time, which is shown here. So again, this is the difference response, so we don't see the visual response anymore, which happens after about 40 milliseconds. We only see the response difference, and this is layers and this is time. And you see that it's particularly pronounced in the superficial layers and in the deep layers, and somewhat weaker, but not entirely absent from layer 4, the input layer of cortex. Now we can also then look at the CSD for this difference response, and then we found something remarkable. So the figure gives rise to an extra current sink in layer 1 and layer 5. Precisely the terminals of the feedback connections. Now this was the situation when the receptor field was in the center of the figure. We also wanted to see what happens if the receptor field falls on the edge. And then the profile of figure ground modulation in spiking across the layers is very similar as when the receptor field falls in the center of the figure, with one exception. Remember that this boundary detection is a very early process, so we should look early in time to get an isolated boundary detection process, and you see that it's actually only happening in the superficial layers, as if the superficial layers have a special role in the detection of feature discontinuities. So let me summarize this part of my talk. So you present the figure ground stimulus to the visual system. In V1, you get driven activity that starts in layer 4 and layer 6. Of course, there are these connections between the different layers, so activity quickly spreads to the other layers as well. And then you have this early process that seems to be happening mainly in the superficial layers. We think it's iso-orientation inhibition that gives rise to extra activity in the superficial layers that then is responsible for boundary detection. Then you have to wait a little bit for the feedback to arrive from the higher visual areas. That happens in layer 1 and layer 5 and drives extra activity, particularly so in the superficial layers and now also in the deep layers. Okay, so we start to have a nice idea of what's going on in the different layers, and indeed the different layers seem to have specific roles during this task, which is why it might be interesting in the collaboration with Maastricht and also with Reiner and with Wim van Duffel in Leuven to look at this profile in high field MRI to see where we can resolve the layers and the activity in the different layers during a comparable task. Okay, now I don't have a lot of time to explain this, but we also did some pharmacology in this task, and we find that the feed-forward part of the response relies heavily on the AMPA receptor, whereas this feedback pathway seems to rely more on the NMDA receptor. Now, the NMDA receptor and the AMPA receptor are both glutamate receptors, but AMPA receptors are really good in activating cells, and NMDA receptors are in some sense more modulatory. And this seems to be confirmed when we started to do these pharmacological studies. Okay. So now in the last 5 minutes or so, I would like to now show you the exception, because we typically tend to think that feedback connections are modulatory. But we got some evidence that they might also be driving in some conditions. So I want to start talking about imagery, and this is something that you cannot easily study in monkeys. Now we have a collaboration with neurosurgeons at the Free University in Amsterdam, and they have patients that go into this epilepsy program, and they get electrodes implanted in different areas of the brain, and it hardly ever happens, but in this case we had a patient that had an electrode in the early visual cortex because the epileptologist thought that this was one of the possible sources where the epilepsy could spread from. So this patient had an electrode in primary visual cortex or area V2, we are not entirely sure yet. What you see here is that these big black squares, these are the clinical electrodes, but there were some small micro wires sticking out here that allowed us to record from multiple unit activity in these early visual areas. And what you see here is the patient from the back. So we have an eye tracker here, so we can instruct her to maintain fixation at the fixation spot. And at the same time, we then can record activity from those electrodes and sort out our filter so that we get the multi-unit activity. Now, the first thing that you then do if you record from early visual areas is mapping out the receptive fields of the cells. And here are the receptive fields. It looks a bit blurred. But here are the receptive fields for different multi-unit recording sites, and here mapped with a different method. And then we asked the patient to maintain fixation at the fixation point. And in some of the trials, there were always these two circles on the screen in some of the trials, and actually this circle coincided with the receptive field. So we then presented a black square either in the receptive field or somewhere else, or we did not present any stimulus. And we also had imagery conditions. So now the patient was looking at a fixation point, we gave a cue, and then the fixation point changed color, which was the cue for the subject to start imagining a black square at the circle that was cued. So here outside the receptive field, and here the subject was imagining a black square inside the receptive field of the cells. And this shows you the responses. I mean, we're still in the process of analyzing these data, so these are kind of our first sweep of the analysis. But what you see here in black is the response elicited by a black square in the receptive field. In gray is a much weaker response by a black square outside the receptive field. This is what you would predict for cells in primary visual cortex. Now this is the imagery condition when the subject was imagining something at the location of the neuron's receptive field. And you see we got quite a response here. And that did not happen if the subject was imagining a black square outside the receptive field. And we also had this condition in which nothing happened. Okay, and this is quantified in this graph below here. So here the subject was imagining something in the receptive field, and here the subject was imagining something outside the receptive field. Okay, so this is an exception where we see that imagery can actually activate spiking activity in early visual cortex. Now we don't know yet whether this is area V1 or area V2. Okay, so let me phrase my conclusions. In the beginning of my talk, I've been talking about this texture segregation task, and there we saw we need boundary detection and region filling, and we seem to find good evidence that boundary detection is an early local process which occurs in the superficial layers, whereas this region filling process depends on the feedback from the high visual area, and it's strongest for figures that are relevant to your task. And it mainly occurs so in the superficial layers and in the deep layers where extra spikes are elicited. We found that feed-forward connections tend to drive cells in the higher areas, and feedback connections tend to influence visually driven neurons. And we believe that we now start to understand how the interactions between those different brain areas are responsible for this process that labels all those image elements that belong to the same perceptual object with an enhanced response. And the last part, I suggested to you that through these human recordings that it seems to be the case that imagery can activate neurons in area V1 or area V2. Okay, now last conclusion slide. So this is a problem that we have been studying a lot. That's the question, how does the visual system label all those image elements that belong to a single perceptual object? And I think that we now start to have very strong evidence that this labeling process is done with enhanced neural activity. These are the people who did the work. So Jasper Poort was responsible for the study where we investigated the effects of attention on figure ground segregation. Matt Self did the Lamro study. The modeling part was done in collaboration with Florian Raudis, Hogebos, and Heiko Neumann. And the multi-unit recording in human patients was also done by Matt Self together with Judith Peters, here from Maastricht, and Jessie Possel. Thank you very much.